Well, welcome all participants to this IAS Educational Fund webinar entitled Research Priorities for an HIV Cure, the IAS Global Scientific Strategy. My name is Sharon Lewin. I'm Director of the Doherty Institute at the University of Melbourne and joining you late at night from Melbourne, Australia. Thank you all for joining us today. Our aim for this webinar is to provide an opportunity to discuss new updates on HIV cure and their implications for people living with HIV, implementing partners and governments. Presentations will also highlight how to implement the new IAS Global Scientific Strategy from 2021 in the current context of health systems and challenges due to the COVID-19 pandemic. I'd like to introduce a few housekeeping rules for the webinar. First of all, we have a large number of attendees, so all participants are currently muted. We've also planned time after the presentations for a Q&A session, so please send any questions that you may have for our speakers, focusing on today's topic in the Q&A box at any time. If your question is for a specific speaker, just indicate their name when typing your question and I'll hopefully direct it to the right person. You'll also notice there's a thumbs up symbol when someone will ask a question in the Q&A box. And if you've got a similar question or find this question important, please click on the thumbs up sign to help me prioritize the questions. And in case you're having any issues with Zoom, you can find additional guidance provided in the chat box. Please also feel free to send us any technical questions you may have there as well. Let us know which country you're connecting from and what time it is for you. We love seeing some interaction in the chat. I'd also like to take a moment to thank our donors, ViiV Healthcare. So it's my very great pleasure to welcome today's speakers, and they include Deborah Persaud from Johns Hopkins University School of Medicine, Steve Deeks from University of California, San Francisco, Simon Collins from iBase in the UK, and Cissy Kityo from the Joint Clinical Research Centre in Uganda. Now it's my very great pleasure to introduce our first speaker, Deborah Persaud. Deborah is a Professor of Pediatrics at the Johns Hopkins University School of Medicine. She's a clinician and researcher who specialises in the study and treatment of HIV infection in children, and she also directs the Pediatric Infectious Disease Fellowship Program at the Johns Hopkins Children's Centre. Deborah will be presenting on HIV cure in the news, stem cell transplantation for a New York woman. So Deborah, over to you.
Great. Hello, everyone. Thank you, Sharon, for inviting me to present this exciting case. And I only have 10 minutes, so I'm going to go very quickly. This is a case of HIV remission in a woman in the US who was enrolled in the International Maternal Pediatric Adolescent Clinical Trials P1107 study. The study was led by Yvonne Bryson and presented this year at CROI. Just to start, Impact P1107 actually has a long history. It was the first impact protocol to try to attempt to assess cure in children, but we opened this study to children and adults living with HIV. It's an observational study, and it was meant to look at the effects of cord blood transplantation with CCR5 delta 32 donor cells. Now, both heterozygous and homozygous donor cells were included, but the participant we're going to present today was transplanted with the homozygous CCR5 delta 32 cells. The overarching goal in 2013 when we developed this protocol was to replicate the Berlin patient. We designed it specifically to use the biobank that existed of previously screened cord blood units. And I'll get into more details of the advantages of cord blood units, especially for racially diverse populations. And we estimated that if we had about a biobank of 300 pre-screened cord blood units, about 80% of participants could potentially find a match using some genetic screening we did here within the US. And this was a collaboration between the Impact, the Bone Marrow Transplant Research Center, and co-endorsed by the ACTG. So quickly, we just be posited that by having access to these pre-screened cord blood units with CCR5 delta 32 gene mutation, we could expand the pool of donor cells for stem cell transplantation. and potentially benefit non-Caucasians of mixed race ancestry, given the widespread availability of cord blood cells, lower stringency requirement for HLA match for transplant, and lower incidence of graft versus host disease. And so as most of you know, and all of you know, to date, there are 2 cases of HIV cure, the Berlin patient in 2009, and then 10 years later, it took 10 years to find a second patient, a London patient. And the shared feature here between these 2 participants are really the CCR5 delta 32 homozygous bone marrow cells that they received, in addition to intensive chemotherapy, and in the case of the Berlin patient, total body irradiation. What I want to point out here that we struggled with our case is how to ascertain that someone has been placed in remission to take them off therapy to actually detect remission. And you can see in the case of Berlin patient, his ART was stopped immediately after transplant, and he never rebounded since his first transplant. And in the London patient, his ART was stopped 16 months after transplant, was first reported in remission in 18 months, and then at 30 months was deemed cured by his care providers. So as most of you know, this CCR5 delta 32 is a rare mutation. There is no routine screening of bone marrow donor banks, but cord blood banks serve as a potential solution, as I mentioned. The main advantage of adult donor cells is you get a high dose from a single donor, and there's rapid engraftment of cells, but the disadvantage is this high HLA match that's required. With umbilical cord blood cells, on the other hand, a major disadvantage is that you can only get a limited number of cells per units. So sometimes cord blood units are combined to get sufficient dose for engraftment. And then there's also delayed engraftment. In our situation, what was used is what's called a combined haploidentical cord grafts, where an adult donor cell is given to support engraftment of the cord blood cells. So this participant received a haploidentical adult stem cell donation in addition to the cord blood transplant. So I'm going to quickly go to the laboratory studies to assess the effects of the transplant and on HIV persistent within our participant and following ART interruption. And these are standard biomarkers in terms of plasma viral load, total HIV DNA levels, the reservoir size, tropism of virus isolates recovered from the latent reservoir, antibody responses, and of course, antiretroviral drug levels to ascertain that the participant really is off antiretroviral treatment. So the case is a woman who identifies as mixed race. She was diagnosed with acute HIV in 2013 and was actually suppressed with antiretrovirals, but diagnosed with AML in 2017. And with her physician, she actually opted to go for this option of cord blood transplantation because 5 units were identified in the stem site cord blood registry. She also had a haploidentical adult donor to be used for the haplocord transplantation. So she underwent this in 2017. This is a very complicated slide, but shown in blue, which is really the hallmark here, is her plasma viral loads. At the time of diagnosis, very high viral loads. And even before she received her transplantation, she had blips in viremia, confirming the presence of HIV virus-producing cells before transplantation. She underwent transplantation and was discharged very quickly from the hospital. And as you can see, she received several vaccinations, including COVID vaccination, after ART interruption, and did not have any episodes of viremia. So what you see here is her conditioning regimen with fludarabine, melphalan, and total body irradiation. She was monitored for 37 months post-transplantation with the biomarkers I described earlier. And then in consultation with her primary care team and herself, she decided to stop antiretroviral treatment and was monitored closely for 14 months and has remained in remission. Since this report, it's 19 months now off antiretroviral treatment, so we would consider her in remission. We have not deemed her cured because we don't really know how to decide on that effect. So notably is really this absence of rebound viremia shown in blue by open circles following ART interruption. And this is a snapshot of her immune reconstitution profile. You can see in green are her CD8 cells, in purple CD4 cells, and in red her B cells by CD19 and NK cells in black. And you can see following her conditioning regimen and chemotherapy, complete loss of these cells, but then immune reconstitution about a year following transplantation. And what's really notable, this is a really nice graphical representation. On the left, you see reconstitution of a myeloid compartment. In blue are the adult donor cells, and in gray, you could see engraftment and complete replacement of the cord blood cells by about 7 weeks post-transplantation. That's maintained up to 4 years post-transplantation. And then on the right-hand side are CD3 positive T cells, and you can see at 2 weeks, some of her cells were still present as shown in orange, but then there was complete replacement by 7 weeks that's maintained. So this is complete immune reconstitution with an umbilical cord blood stem cell transplant that is CCR5 delta 32. So following transplantation, what we also observed is marked decrease in immune activation markers. As you know, immune activation and persistent immune activation is a hallmark of HIV infection. And so post-transplantation and also post-ATI or ART treatment interruption, she maintained low levels of immune activation, and she also had no detectable HIV-specific T cell responses in peripheral blood post-transplantation. This is a complicated slide, but each panel represents different viral biomarkers that were measured in her. The filled-in symbol represent that with each marker before transplantation, we could detect HIV-infected cells, a replication-competent reservoir, DNA measured by 2 LTR circles, and plasma viremia at low levels before transplantation. But following transplantation, all of the open symbols represent non-detectable biomarkers of HIV persistence. You can see she did have evidence of LTR circles detected intermittently, which is an HIV DNA molecule, suggesting that not all HIV-infected cells have been cleared following transplantation, even at 14 weeks post-ART interruption, but detection of DNA did not signal impending treatment failure. Notably, with her post-transplantation, she's lost all HIV-specific immune responses by antibody responses, and you could see by the dark bands is reactivity to HIV proteins that have been lost, suggesting really lack of HIV antigen exposure in this participant following transplantation. And then when we tested her cells, shown in red, her engrafted cord blood cells for susceptibility HIV using laboratory strains R5 and X4 variants in the top panel, and her own 2 latent viral reservoir isolates were recovered pre-transplantation. Her cells are not capable of supporting HIV replication, whereas in parallel testing with the donor blood cells, those cells are capable of being infected, again, confirming resistance to HIV, supporting HIV replication post-transplantation. And so to kind of summarize, in the New York patient, this is a woman of mixed race who was diagnosed during acute HIV infection, had great responses to antiretroviral treatment, except for a blip with low level viremia when she was diagnosed with AML, when her antiretroviral treatment was stopped transiently for neutropenia, who had ARVs detected in the post-transplant period, none detected in the post-ATI period, with 14 months of aviremia off antiretroviral treatment that heralds remission. And as I said, she's now 19 months off ARVs and remains in remission. So to summarize, as I said, the first woman of mixed race, the 2 participants reported are males, a person living in the US, a woman, transplanted with haplocord stem cell transplant, with actually remission of 2 diseases, her AML now, and also HIV. We have no detectable replication competent latent reservoir so far, with over 75 million cells cultured. With remission, there's a complete absence of HIV-specific immune responses. There's transient detection of HIV DNA, which likely suggests that there's still trace cells, participant cells still present, and she has no graft versus host disease. We believe this case provides hope for use of cord blood cells and haplocord for remission in individuals living with HIV who require transplantation for other diseases. It's an additional proof that reservoirs can be cleared sufficiently to afford remission and cure in the setting of resistant target cells. I want to acknowledge this is a heroic effort over a decade. The protocol chair, Yvonne Bryson, I'm the scientific committee chair for curing protocol virologist. The team that cared for this participant and discussed her transplantation and manages her, Jingmei Su, Kyung-Ram Leeson, and Marsha Gillespie at Weill Cornell Medical College, Semstide Biobank, and all the laboratory teams involved in this. Thank you.
Thanks very much, Deborah, for a really fantastically inspirational presentation of a great case. We're now up to about 520 participants on the call, and I can see people coming in from all over the place: Oman, Uganda, Cote d'Ivoire. Adam Castillejo is on as well, I saw, from London. So kind of exciting to see the community coming together at this webinar. So it's my pleasure to introduce our next speaker, Professor Steve Deeks. Steve is Professor of Medicine in Residence at the University of California in San Francisco. He's a world-renowned expert on HIV-associated immune dysfunction and its impact on the viral reservoirs. He also maintains an active practice in primary care clinic for HIV-infected adults. And Steve was a key leader in the recent publication from the IAS on research priorities for an HIV cure. And he's going to take us through the key points from the IAS Global Scientific Strategy. So welcome to Steve.
Thank you, Sharon. I'm loving this chat. I mean, I think we have participants from basically almost every country in the world. So this is quite exciting. I need my slides. So what I'm going to do, once the slides show up, is walk us through the most recent version of the International AIDS Society Global Scientific Strategy. And just, first of all, a bit of a history here. So back in, I think, 2011, Francois Barré-Sinoussé and Sharon Lewin and some of the leaders from the International AIDS Society, we assembled a team. This is back when the whole HIV cure thing was beginning, to put together sort of a roadmap where to go for an HIV cure. It was quite successful. We updated it in 2016, and that also had a great impact. And so once again, over the past year or so, we assembled a new team this time to come up with an update of those recommendations and try to plan out a roadmap for how we're going to get to an HIV cure, focusing on the work that needs to be done over the next 4 years. So how did we go about doing this? The process is very important. We really wanted to make sure that this was representative of all the major stakeholders in the world, particularly in ensuring that we had engagement from both the community as well as scientists from various different countries across the world. So what we did was all online, unfortunately, because of the pandemic. But we assembled a group of experts, generally bringing in some new, younger investigators this time, identified 8 major topics, 8 working groups. These working groups met a few times. They came up with recommendations. These recommendations then were passed on to a steering committee, which included Sharon and myself and the chairs from each of the working groups, as well as Simon, who's going to be presenting after myself. And we put together a draft that we then went out and aggressively socialized and made sure that most stakeholders that we could reach had an ability to sort of contribute to this. This was done online and through one-on-one interviews with key opinion leaders, and essentially came up with a document that we're all quite proud of, and it was published, I think, World AIDS Day in December. And these are the groups. I'm going to go through real quick the greatest hits in terms of what we think we need to be doing for each of these areas of investigation. This time around, we took a bit of a different approach. We summarize what's happened in the past 5 years, both the successes and the failures, and then based on that, identified sort of what the critical issues are today. And then, of course, most of the work in this document is focused on the next 5 years, what we internationally, globally need to be doing. So what is that? All right, so again, we'll go quick, but I won't get too technical here. First, we need to understand the nature of the reservoir why it persists. And this figure, if you look in the inner circle, right, there's really a lot of interest right now in trying to determine the landscape of the provirus. Where in our chromosomes does HIV reside, and does it change over long-term therapy? Lots of fascinating data coming out suggesting that the reservoir, that HIV that exists in our body during long-term treatment, actually is not stable, and it evolves in quite dramatic and very important ways over a period of 10 to 20 years. We need more information on that. We need to characterize which cells are infected, what they look like, how they can be identified, what tissues harbor the virus, and so forth. And then there was a really sort of a clearly a need to understand precisely why these cells, these HIV infected cells, which can persist for decades, why they don't die. So this is sort of a kind of our general approach to sort of understanding the reservoir. Obviously, as we get into the point of doing clinical trials in HIV cure, we need to understand with much more precision how to measure the reservoir. We're making some advances here. It's technically been proven to be quite challenging to come up with a blood test that will identify how much virus is in the body, what it looks like, whether it can rebound or not. And so we need to continue to pour tremendous amount of resources into coming up with basically an equivalent of the viral load test that we're all familiar with. You know, a quick blood test that can be done anywhere that will tell us how much virus is there and how much we have to worry about. Has to be high throughput, has to be likely is going to have to actually quantify where in the genome the virus lives, because some of these viruses are in sort of the graveyard of our genome and probably doesn't matter. Other are in active genes, and those are probably the ones that we need to worry about. Clearly, we need to do a better job of actually coming up with assays that measure all the subtypes. Essentially, not too surprising, most of the current assays focus on subtype B, but everybody is in agreement that all these assays now need to be defined and expanded to make sure that we can deal with the global pandemic. We're quite interested in developing assays that will measure not just the amount of virus, but whether that virus is making protein, because it's protein is what therapeutics typically go after. And that's actually proven to be quite difficult. And personally, I think probably the most important thing we need to do in this realm is come up with the ability to test the viral load or the amount of virus that people have after stopping antiretroviral drugs in real time, at home, as frequently as a person needs to do so. Because in these cure studies, and presumably once we have a cure, if we're not 100% sure the virus is gone, people are going to need to know on a day-to-day basis whether or not their virus is there or not. So a lot of focus on developing measurements for the reservoir. You know, some of the earliest clinical trials in this area have sought to look at this issue of shock and kill, forcing the virus out of the cells so that the immune system can kill it. We've made some progress. We believe, do this, certainly in monkeys, probably in people, but we're nowhere near doing it to the level where it's going to be clinically significant. More work needs to be done in this area, but also more work needs to be done in the opposite area, which is if we can't get it out, can we push it back in so deep that it doesn't come out ever? So this is the block and lock approach. And clearly we need to have more focus on developing this novel approach to the reservoir. We're all familiar with this concept that maybe 1 in 10 plus or minus of people who go on antiretroviral drugs early, stay on therapy for a few years and stop. That 1 in 10 of those individuals will have only a small rebound. These are called post-treatment controllers. They imply that treatment can change the relationship between the host and the virus. We have no idea why this is happening. So we need to understand, we need better mechanistic work in the animal studies. And we begin to need to define which parts of the immune system is actually being modified by early therapy and increasingly by some of our curative interventions, which part of the immune system is actually the one that's actually having an impact on controlling the virus. And so there is a global effort right now to assemble more of these post-treatment controllers and to do this deep dive. Shifting now from more of these basic science studies to actually interventions. Most of the first generation of clinical trials that have looked to cure, quote unquote, cure HIV have used various forms of immunotherapy, basically putting people on antiretroviral drugs and then doing everything we can to build up their immune system so that then we stop the immune system and control it. Huge amount of interest and effort right now in developing better therapeutic vaccines. We believe that the work in COVID, which was built, by the way, on the work in HIV, but we actually believe that the work in COVID will now provide insights into how we can do this. I don't think anyone believes that a vaccine by itself will build up the immune system sufficient. So we need to identify what else to give: vaccines, antibodies, CAR T cells, latency reversing agents, adjuvants. There's a whole laundry list of various things that can be combined to result in basically the ability to control the virus after people stop their drugs. Great success in monkeys. I'm optimistic that we will actually soon be seeing success in people. That success, I'm going to warn you, will come at the cost of people having to get really complicated interventions that probably no one's really going to want to do when they can take one pill once a day. But the hope is that once we begin to have success in people, all these approaches, which are complex right now, will be optimized and simplified. I think probably a lot of the growth in the HIV cure world will be in gene and cell-based therapies. There is a huge amount of advances being made elsewhere in medicine, particularly diseases like sickle cell disease, cancer, even heart disease, and so forth, in which we are coming up with gene modification strategies that will allow us to potentially cure HIV in the way that we did with the Berlin patient and the New York patient, and I guess our guest honor in this webinar, the London patient. And what happened in those cases that Debbie just described was not the kind of thing that's actually going to be scalable at all. Those transplants were done to cure cancer, not HIV, but they provide inspiration and a clear roadmap to come up with ways in which we can do this in people. And there are two broad ways of doing these gene therapy approaches. One is that you take the cells out of people, take it to the lab, and modify them. And these studies are ongoing right now, and we're making cells into killer cells, like CAR T cells and so forth. But I think the next generation of work that will need to be done, it's going to take a lot of work to come up with a way to do this safely, is to actually not have to take cells out of the body and send it to a very fancy lab, but rather, theoretically, I think it is possible to bring people in, if they're on treatment, give them a shot in the arm, and then whatever that shot does, provide some kind of transport mechanism, a viral vector, something else, that will then take the gene modification approach directly to the T cells throughout the body and cure. Huge amount of work is going on this throughout biomedicine right now. And people like Cissy, who's going to be speaking next, have been really just passionately trying to take all this great work that's being done largely in rich countries to low and middle income countries. And I'm sure she'll be talking about that work. So a lot of great excitement in the broad area of gene and cell-based therapies, major focus over the next 5 to 10 years. We didn't have pediatric cure in the past 2 editions, and Debbie gave us a hard time about that. We fixed that problem, and we have a whole section on what needs to be done in terms of coming up with a cure for babies and children. A lot of the approaches are similar, but there are very important distinctions that are outlined in this, in our document. And finally, I mean, we all know, and I bet Simon will talk about this, we all know that, you know, that we can come up with these fancy cures and gene therapy and cell therapy and vaccines and all sorts of stuff. But if we don't prepare the field for these therapies, they're not going to be taken up. We need to know what all the various communities with HIV want from a cure, what they don't want. Will they take it? Will someone pay for it? And so they're really, it's increasingly becoming a strong focus of everything that we do in HIV cure world, is making sure that we understand the ethics of what we're doing, social implications, and making sure that we actually are engaging in a real way from the beginning of people living with HIV in all the various different types of communities. And so this is an absolutely critical part of what we're doing, and a whole section is devoted in these guidelines on how to get there. I will say that the International AIDS Society has been leading efforts on multiple levels to make sure that all the cure work, most of which is being done in places like the United States and Australia and so forth, that much of that work will be applicable to the rest of the world, but not all of it. And there are a very important list of questions that are going to be very specific to low and middle income countries, particularly sub-Saharan Africa. And this concept is basically described throughout our guidelines. Finally, just to warn people, you're going to hear about various different interventions that we think might help. And I believe we're going to begin to make incremental progress, probably on anecdotal cases, with interventions that no one in their right mind would ever want. Because, you know, again, we have pills and we have injectables. But just like it was with antiretroviral drugs back in the 90s, once we identify interventions that work, then optimizing them iteratively will basically become critical. And the IAS, working with the Bill and Melinda Gates Foundation, efforts that Sharon and I are very much involved with, are coming up with, you know, descriptions of what it is exactly that we need to deliver, how we're going to get there, and what's going to happen with these various different generations. So a lot of science in 15 minutes, and happy to take questions. Back to you, Sharon.
Thanks so much, Steve. Great overview of a complex, detailed document, but also injecting a bit of realism into what people can expect, which I think many of our audience will want to know about. A reminder that the actual full document's available online, open access through Nature Medicine, and also there's a recording of all the presentations that will be available to all the participants. It's my great pleasure to introduce our next speaker, Simon Collins. Simon is an HIV treatment advocate who co-founded iBase in 2000. He's involved in developing community involvement in clinical research and treatment guidelines. He's also collaborated on generating non-technical information on new advances in science and on the impact of COVID-19 on HIV. And Simon was a part of the working group that Steve just described, a fantastic contributor, and guiding us in how to interpret the complex science for a broader audience. And so Simon's title of Simon's presentation is the IAS Global Scientific Strategy 2021 into Action Advocacy Framework. So over to you, Simon.
Hi, thanks, Sharon. It's fantastic, lovely introduction, and amazing group here that IAS has pulled together. Really fantastic to see everything coming through. I haven't got slides yet, so if they're going to be shared. Yep, this looks good. And thanks also for including the community in this webinar today. We've been included one way or another from the beginning of all the IAS work on cure. And that's really appreciated as well. No disclosures. And so just for 10 minutes, I'm going to do a brief outline of community involvement in this strategy and the document that was published in December for World AIDS Day, and just talk about a few of the community-related issues that came out of this document, and going forwards as well, how we can have broader engagement of community. So this year was the third of the IAS 5-year reviews, and I think it's important to say that it's an incredibly condensed document. It's the main paper is very technical, very scientific, and it compresses results from over 170 new papers since the previous review. And the writing group includes more than 40 experts globally. I haven't got a picture of them, but I put a picture of both Steve and Sharon here because without that level of commitment to community involvement at a leadership level, it wouldn't happen. And this has been remarkable all the way through. And then, as well as making sure that there were community reps on each of the chapters and on the main writing group, we also worked collaboratively with a group of about 20 advocates from the US, Europe, Southeast Asia and Africa. And there might have been someone from Australia there too. So this year we produced 3 versions. The first is the main paper in Nature Medicine, which is it's a document really everybody should look at, but it's fairly technical. If you don't have a technical background, then you're going to maybe have a little bit of help or support in terms of what some of these sections are talking about. And the IAS also produced this really helpful summary for medical journalists. This was translated into, it was available in English, French, Portuguese and Spanish. And so I'd recommend going there. And then we also, the writing group, the community reps of the writing group produced a very simple, non-technical Q&A in everyday language, explaining what the main document was about. And it was highlighting the advances in the last 5 years and what we were hoping to achieve in the next 5 years. It's written at a reading age of about 10 or 11, which makes it very easy if you want to just cut and paste bits for translations in newsletters. And so hopefully that's useful. It's important that all these documents are available as open access and free online. So some of the key points that came out as being specifically to the community reps in the group included broadening the awareness and involvement in cure-related research. And it's important the language we use cure-related rather than cure research, because sometimes that gives a much clearer indication that we're just looking at each study is maybe only looking at a section of cure-related work. And although there's lots of advocates, I work as a treatment advocate, I've seen lots of other people signing in here, work for HIV organisations, and we do a lot of work in trying to raise the profile of this research, but we really want more involvement. We want broader support. We want this to be an easier thing for everyone to be involved in. There were a few ethical issues that I'm going to talk about briefly, and importantly, we want that any eventual cure really is going to be accessible for the majority of people living with HIV. So wider community of awareness, and there was a sentence that talked about this in the main document, and it highlighted the fact that current engagement is suboptimal. And it always will be, really. We're excited by all this and we want everybody else to be excited too, but there's certainly bottlenecks where it becomes difficult to follow the research. And so initiatives like the IAS paper will hopefully make that much easier. And although community networks and advocacy networks are sometimes brought together to support individual cure studies, with community advisory boards. We want the awareness to be out there in the broader community across all demographics and populations that are affected by HIV. And really, because that's because it does affect us all. At some point, everybody who's HIV positive and living with HIV will wonder about if there'll be a cure. Will there be a cure in my lifetime? Will there be a cure in 5 years' time? We want to know what that will involve. And actually, we want to be involved in shaping this research. And there's been a couple of really useful surveys of people living with HIV. And sometimes what we're looking for from the research and from the cure is a little bit different to the thoughts and the ideas that some of the researchers are working on. And so we like to bridge this gap, bring everyone together, make sure we're all talking about the same thing. So broader inclusion in studies is one of the other issues that sort of came up. And I've referenced a study down here by Liz Barr and Richard Jeffries. And they've done a couple of reviews where quite often cure-related research doesn't even report the demographics of the people involved. And when they do report the who's being studied or the participants, they tend to be in high-income countries, predominantly male, predominantly white men, often gay men. And actually, we want this even basic level research to have broader inclusion by age. Steve talked about the importance of adults and children. There's different issues going on there. Children technically might be living with HIV and treatment for 70 or 80 years. So arguably their need for a cure is probably better than, or it's just as important as for adults. And we want the inclusion to reflect global need, not just as a question of equity and inclusion, but because there's good scientific questions there relating to viral diversity and different HIV subtypes. And then the research that's funded to look at some of the biomedical aspects of cure-related research can also be used to find out what participants are thinking and how they feel about this research. And so including social and behavioral research is also important. We're interested in the real cures. We understand that these aren't going to be interventions that are going to be available to most people. Most people wouldn't want to go anywhere near the things that are involved in the few cases of cures that we have. But we're interested in these cases. Pointed out that Adam's connected and is in the webinar today, and that there's also different approaches to clearing HIV that we're also only finding out about. So I thought somebody else in the chat referred to Laureen's case, where the viral genome was maybe in a silent area of our DNA, and that's really important to understand that. And the case, going back even further, the cases of Visconti cohorts, which are not cures, but they allow some people to have long periods without HIV treatment. And for lots of us, that would be very similar to being a cure. So some of the ethical questions that came up, one of the common ones, it's been reported quite a lot, is that there's a disconnect sometimes between researchers and participants, even when the researchers go to a lot of trouble to emphasize that no one will be cured from this particular piece of cure-related research. It's quite common to then speak to someone that's enrolled who suddenly, who thinks at the back of their mind, well, I might still be lucky in this study and end up being cured. And that's more than an anomaly. It's something that has to be taken really seriously, because really it voids the informed consent for that person that's in the study. If someone thinks they might be cured in a study and there's no chance of that, then that disconnect's not. And then there's also issues about having to, we have to stop, have a period of stopping treatment in order to find out whether a cure has been successful or not, and there's ethical issues related to treatment interruptions. Quite often that focuses on the risk of sexual transmission to the partners of someone who's taking a treatment break. But there's also been a case reported of the HIV positive participant becoming reinfected during the treatment break because this was something they hadn't been aware of. It's really important that we use appropriate language, that somebody going into one of these studies isn't going into a cure study, they're going into a cure-related study. And so these ethical issues, again, the community will work collaboratively with researchers to try and move the field on. Essential to any success we have in cure research means that it's going to have to be available globally, not just as an afterthought. There's already some groups working on this. There's an initiative called AgeCap, which is looking at accelerating access to cure-related achievements, really, in Africa, but other groups are thinking about this too. And really, to be really successful, it's going to have to be something that follows the global need, really. I put in a slide about cure awareness as treatment literacy. So lots of advocates take a lot of time to explore things like your CD4 count, your viral load, talk about drug resistance, importance of adherence, and actually awareness of the science behind cure is just as an important section of treatment literacy. First of all, you've got someone's attention because ultimately that's what we hope is going to happen, and we hope at some point we might be cured, but also it explains adherence and resistance really are very closely connected to this viral reservoir. And so as an active part of learning about your health and your HIV status, knowing that there's a reservoir, knowing that it comes back within probably a few days if you miss your treatment for a while, the whole thing sort of connects together. And sort of the importance of cure-related research, I guess, is it gives people optimism, and it's always been a hope. Even long before there was treatment, there was always a real hope that we'd find a cure at some point. I put the same picture in here that Steve put in. I think graphics are a good way to make points that would otherwise take a page full of text. And for treatment literacy, it's useful to know that even though we're being monitored based on a blood test, like a surrogate marker for how well treatment's working, that actually the scientists and researchers are concerned about HIV that might be in small pockets in different parts of the body. The question that everyone would ask if 1,000 people on this, or 2,000 people maybe on this webinar, would be when is a cure going to come? And of course, that's the question that nobody's been able to answer, although it's always been about 10 years away, like an HIV vaccine. And I've been sort of an advocate for well over 20 years, and the vaccine's always been 10 years away. Cure has always been about 10 years away, but I think we could be more confident that it's a closer 10 years than it used to be. So there is real progress that's being made. There is much better understanding of what the challenge is, the scientific challenge. I suppose in a summary that anything that gets us to the point of being able to stop treatment without needing art and without being a risk to our partners is going to likely have several stages, approaching the reservoir and then boosting our immune system. And Steve really rightly mentioned the importance of being able to monitor this at home with the equivalent of a home-based viral load test. This is the people who were involved in the community group. I'd like to thank them for all their support. Everybody was active, everybody got engaged and appreciated the chance to be involved in such an important document and program. And I'd like to thank you and happy to take questions later.
Great. Thank you so much, Simon, for taking all that complexity and making it much more simpler for a largely community-based audience that we can see out here. And also, again, just noting we've got over 600 people now in the webinar, and lots of colleagues and friends from across Asia Pacific are coming in from Cambodia and India, Pakistan, the Philippines, and great to have all of you here with us. So great pleasure for me to introduce our final speaker, Dr. Cissy Kityo. Cissy is a medical doctor, a public health specialist and a researcher. She has a master's degree from Johns Hopkins School of Public Health and a PhD from the University of Amsterdam. She's currently the Executive Director of the Joint Clinical Research Centre in Uganda. She's among the pioneers of antiretroviral therapy in sub-Saharan Africa, and once again, being a pioneer in HIV gene therapy in Africa. And Cissy is going to speak to us today. Her title of her presentation is Global Research Collaboration for an HIV Cure, the Way Forward. So welcome to Dr. Kityo.
Thank you, Sharon. I take this opportunity to thank the organizers of this meeting for inviting me to give an overview of the global research collaboration for an HIV cure and what is our way forward. So, Global Village is clearly divided between the Global North and by World Bank and others, and some of the divisions are based mainly on socioeconomic status. But there are also other differences between the Global North and Global South, and these give us a premise on which maybe we can leverage to support access to HIV cures across the world. So there is differences in disease prevalence, in particular HIV. There is more advanced healthcare infrastructure in the North. There is more national dedicated research and development support for health in the North. And then there is presence of major market players, including pharma and funders for research and development. And the genetics also is different, especially in sub-Saharan Africa to other parts of the world. And then historically, high-income countries have supported low and middle-income countries to control the HIV epidemic this far. So we really hope we can continue this partnership and collaboration to bring HIV cures to developing countries. This slide just shows we have about 38 million people who are living with HIV today, and the majority of those are in low and middle income countries. And mainly 67% of this 38 million is in sub-Saharan Africa. Despite the fact that we have very potent antiretroviral drugs that we use today, HIV/AIDS remains a leading cause of death. There are also many people who are not able to access treatment, and many of these are in sub-Saharan Africa. And I think we see there is, we are starting to see is what we would want to see across the world is a cure which can benefit 38 million people living with HIV today. So when we talk about HIV cure prospects, this presents a very promising new HIV management paradigm. We are optimally looking at a one-time treatment with a single dose or injection. We are looking at cure of HIV. And then we know that if we have a cure, there will be very high impact on quality and quantity of life with great clinical and economic impact, plus social value. So I'm sure all people living with HIV would love to see an HIV cure, and Simon just said it's 10 years away, maybe shorter. However, the Global South is currently excluded from gene therapy development. This is a slide taken from Kenneth Coneta's publication, which is very recent, 2022. And as you can see, the majority of the population is in Africa and Asia. but this is not where gene therapy trials are taking place. And most of this gene therapy that is taking place today is looking at oncology, at very rare diseases, at neurological disorders, and very few of this is looking at infectious diseases, which includes HIV. Again, from this same publication, you can see that by region, the majority of the studies are happening in United States and Canada and Europe. Africa, we have one study that has been done. It has been done in South Africa, and it's looking at hemophilia. So we have a big gap to catch up with our friends in the global north. Now, this is just looking at the income rating by World Bank. And again, I think you can see high income countries, very many gene therapy studies. And in low income countries, we have 0. This is just looking at some of the upper middle income countries, and you can see China, Brazil, and Turkey. They have a few studies happening. And the one happening in South Africa, as I said, is looking at hemophilia. So this is the situation today. What should we do? Continue lamenting? No. We have to aggressively, from the low and middle income countries, work together and forge a way forward in which we can catch up, because the disease we are talking about today, HIV, affects us most, not where the trials are taking place today. So the differences I already mentioned on the first slide, I think require that we partner. We know our weaknesses, we know our gaps in the low and middle income countries. So we need to forge partnerships, HIV cure therapies. In 2019, the Gates Foundation and NIH joined forces to address HIV and sickle cell diseases, both in the US, but also in sub-Saharan Africa. And as you can see at the top, the pipeline of what has to be done is not short. We have to go from basic research, applied research, product development, clinical trials, and then eventually access. So this is not short, but I think as we did in COVID, maybe we can shorten some of these processes. So NIH and Gates Foundation have engaged pharmaceutical companies that already have expertise and infrastructure, and technology companies to address the basic research and come up with products that we can eventually evaluate both in the north and in the south. And this slide is borrowed from Mike of the HIV Frontiers. HIV Frontiers Program is a program within the Gates Foundation. It has laid down groundwork to reach a bold new goal. I think all of us have to be bold in what we propose and what we do if we are to get anywhere. So the goal is that in the next 10 to 15 years, we should achieve effective, long-lasting, and safe single-shot cure. Research platforms have emerged as front runners, and Steve talked about different strategies. They are all going ahead, but these are the front runners today: gene therapy and therapeutic vaccination. And either platform could lead to new treatments with a target product profile, including a single shot, being effective in lowering the viral load, preventing reinfection of treated individuals, being safe, but also affordable. And these research platforms may provide cures not only to HIV, but also for other diseases as well. So we can leverage these platforms when we have them. The gene therapy platform is building on what has been done in ex vivo gene therapy for sickle cell disease. And also, I think Steve mentioned, at this stage, we are at ex vivo, but we aim to go to in vivo, a single shot. The therapeutic vaccination is leveraging the new technology that has been used to produce vaccines against COVID. So we have, I think, as Simon said, we are closer in the 10 years than we were much earlier because we have platforms that we can leverage. HCAP has been mentioned. It's working towards bringing curative interventions into sub-Saharan Africa. And the objective is to develop and ensure the widest possible access to a cure for HIV, specifically in sub-Saharan Africa. So we have partnerships that we are working with, and we should leverage them, and we should make bold moves and bold goals. So in response to this gap, we have founded what we call the Global Gene Therapy Initiative Working Group in 2020. And we are working towards enabling access and implementation of gene therapies as curative medicines in low and middle income countries, initially focusing on HIV and sickle cell disease. The GGTI works in many ways, including advocacy and fundraising. And we know that from 2017, there have been approvals of gene therapies in the US and Europe. And the goal of GGTI is that by 2024, we have phase 1 clinical trials initiated in 2 countries, Uganda at the Joint Clinical Research Center and India. And this is just a start. So the co-founders of GGTI include myself and Professor Jane Adair, who is based at the Fred Hutch in Seattle. And we have built a network of over now 60 experts in gene therapy, in immunology, in manufacturing, and all these working together have made very steady progress towards our goal of having the phase 1 trials and being part of research and development of gene therapy in low and middle income countries. But the emphasis is that patients and advocates are at the table. And these are among the few advocates we work with. I think I saw Moses Charger, Supercharger on the call. Simon, you are welcome to join us. Because right now we are trying to figure out how do we communicate intelligently to the patients and the community so that they understand what we are saying. And also, Adam, you are on this webinar. We would be very glad to have you at GGTI. So GGTI, how are we doing it? And we are not the only ones addressing this gap of access in developing countries, but we are making a dent in this gap. We have identified 4 pillars that we are working on concurrently to be able to have at least the phase 1 trials. And again, we cannot overemphasize collaborations. We already said we do not have the technology, we do not have the funding. So we have to have the collaboration to get transformative clinical gene therapy research. And these 2 members of GGTI include Boro, who has developed an HIV dual CAR T cell product that we are working with, and also John Tisdale from the NIH working with his team. They've developed a product for sickle cell disease and they are licensing it to Caring Cross where Boro works. So Caring Cross is an NGO And really what we are doing is how can we have affordable products to evaluate products that could be afforded within our environments and settings. These two ladies, Els and Jen, are developing the in vivo products that we may also use in future. Right now, what is available is ex vivo, but these ladies are working on in vivo products that may also be used within the GGTI. So what are we doing to access these advanced therapies in low and middle income countries? This is the product that I've talked about, developed by Caring Cross. Steve, who presented earlier, is a principal investigator at University of California, San Francisco, where this trial is taking place now. Within the GGTI, we have agreed this product will be given to us at Joint Clinical Center. And then we are also talking the AIDS that is funded by NIH to fund the clinical trial. And so one of the main cost drivers, and also for which we don't have infrastructure, is manufacturing process for these products. The gold standard is what you see there in the green band. This is the Middleincome produced, this is the Prodigy produced by Middleincome, very expensive, and the policies around it is really not to take this to low and middle income countries. So we are working, and in our group, including Jane and Boro, are working on alternative processes to manufacture gene therapy product. We are also working on a GMP facility, which is a container which can be taken from one clinic to another. So we are trying to see how can we make this cost effective for our settings. And then we are looking at infrastructure, institutional voids and ecosystems. And one of these is we've done one trial in South Africa, I already showed you. So we need to train a team, a core team of people to help with gene therapy in developing countries. So we have some funding from Gates Foundation. Our first trainee is now at Fred Hatch. We have another batch that will be going out, and also others that we have assembled. But these people, when they train, they will become a core for training in sub-Saharan Africa, so that we do not have to send out people all the time to the US. Policy and regulations and guidelines. We don't have them in our countries for gene therapy because gene therapy hasn't happened. But we are working with WHO. WHO has already developed guidelines and the frameworks for gene editing, which we are going to adopt. In our countries, we have to work with our regulatory organizations. We have to work with politicians for policies. And I think those people who are coming from Uganda, you have seen how our minister for research, innovation is being battered. But that one is for us in Uganda. So we have to know how to communicate to politicians what we want. We need to be able to advocate for funding, because we are really looking after patients whose responsibility would be our governments. So this is a big area in terms of communication and also putting policies and guidelines in place. Sorry, the slides are slow and they've been cut off, but I will stop here. And I thank you for listening to me. I think we have a lot of work ahead of us, and every one of us has a part to play. Thank you very much.
Thank you, Cissy. A lot of great excitement with your vision and passion, and a lot of support from the chat to the brave steps that you're taking and being supported in partnership across the world. So thank you for a great talk. I will get everyone to turn on their videos and their microphones. All of our speakers are now open to taking Q&A. I've been trying to keep track of the numbers of lots and lots of questions coming in, but we've got a few kind of strong thematic questions that are coming through. So excuse me if I'm not going to ask your specific question. I'm going to cover some of the main themes. So I'll start with some questions, Deborah, about the San Francisco and Esperanza patient. Simon mentioned these two cures as well, and I think there's quite a lot of interest just to understand what those two cures mean. So Deborah, do you want to just quickly tell us about the San Francisco and Esperanza patient?
Yeah, I think, yeah, you're right. There were comments about why not include those two participants. They're two women, obviously. I think what we were, in presenting this case, we were trying to focus mainly on an intervention that led to remission and cure. And so in those two cases, as I understood them, these were spontaneous control of HIV without any interventions, therapeutic interventions. And so that's why those cases weren't included. But I think to round it out in the future presentations, to kind of say there are other cases of cures, but those cases were really not through an intervention. It's really clearance of intact proviruses, perhaps very early on, and integration in non-permissive sites for the virus to reactivate. And so those individuals are able to control off antiretroviral treatment. I should know this, but I don't think that either of them carry the alleles that confer, that are associated with, you know, what we typically think about as elite controllers. I mean, certainly the Esperanza patient did not, but I'm not sure about the San Francisco patient, whether she had elite control genetic markers.
Yeah, I can't answer that. But, you know, sometimes people use the word of the natural cure, so there's no intervention, and both of these patients had no intact virus, so no complete virus, and seem to have got rid of that intact virus by themselves. Just staying on sort of, again, I think it's an important area to clarify, and we have a question here, but and maybe Steve might tackle this on terminology. Cure or remission, what are the real differences? And is any of this work going to prevent reinfection? How important is that in the cure effort? So that's sort of summarizing a few questions that are in the Q&A. Steve, do you want to give your answer to that?
Yeah, we've been debating these terms for 10 years. I think there's consensus now that remission is essentially the virus is there, we can see it, people are antibody positive, but the immune system isn't controlling it. Elite controllers, post-treatment controllers, some of these therapeutic interventions, they're already creating these sort of situations of long-term remission. You know, it's not ideal. People have the virus, there may be ongoing inflammation, there's always a chance it'll pop up. That's why this at-home testing is just so critical. But we're really interested in remission because we think it's going to be a lot easier to get a remission than a cure. Now, ultimately, and many people may choose to be on treatment rather than have a remission, but it'd be nice to have that option. And a remission will largely be defined as having a viral load below that level where you can infect others. That's critical. And that's close to being undetectable. A cure is the holy grail. We all want a cure. A cure basically means no virus. And typically people become antibody negative and they go about their life and they don't have to worry about it. No stigma, no residual effects, and so forth. That's going to be much, much harder to achieve. We probably have achieved it in the Berlin, the London, the New York patients, although the problem with a cure is you actually have to prove the virus is not there, and proving a negative is really not easy to do. But anyway, so those are the two things, remission and cure. Remission will be easier and maybe not necessarily what everybody wants, but some people will take it. A cure is ultimately where we need to go.
What about the issue of reinfection? So will these cures prevent people from becoming reinfected?
you know, some of the approaches we're taking will absolutely have no impact on reinfection. Whereas others, particularly those in which we're either building up an immune system like Adam's that has cells that are not infectable for the most part, or building up an immune system that's so powerful in controlling the virus it also prevents the infection. I would say half of what we're doing will result in protection against reinfection and half will not. From a public health perspective, it really is going to be important to come up with something that prevents reinfection.
Great, thank you. There's a lot of questions here about community engagement in cure trials, particularly in low and middle income countries. Maybe I'll get Cissy to speak to that, and then also Simon. Cissy, how can we do this, get better community engagement in low and middle income countries by community?
Thank you, Sharon. And I think this is a big question and we need input from everyone. Because I can sit here as a scientist and I talk about communication to the community. We have developed a communication document or brochure. And when we gave it out to the community, they said, this is very difficult. And we thought we spent a lot of time making it very simplified. And they said, this is very difficult. But when we talk about community, we shouldn't generalize. Community is wide. There is community of healthcare workers. There is community of other researchers. There is community of regulatory authority, or regulatory organizations. There is community of politicians because we want them to pass our policy. So community is varied. Who are the stakeholders we need to focus on? So we need support from community leaders, community communication specialists to help us to craft messages and also strategies on how we should communicate. I think for us, we have our role and we think we can do everything. No, we can't. We need support. So that's why I was inviting my friend here, IBES. I read his summaries on internet, simplified things, very easy to read. But so how we get to communities, what messages do we give to communities? We can sit in our offices and think about what to do, but we need support from those communities to advise us. That is why we have an ICAB as part of GGTI, because they are representatives from community and they are also helping us and guiding us on what to do. But I think we can have more varied representation because the communities are really varied.
Yeah, Simon, do you want to talk about that? And maybe, you can comment perhaps on some of the initiatives in the IAS. I could do that as well. But Simon, how do you think we can really accelerate community engagement, particularly in low middle income countries? I'm getting questions here from Pakistan and from West Africa and other places where they say there's no cure research even happening.
So I can think of a couple of things. Usually HIV is still such a psychological knock that every country will have some level of peer support and peer organizations where HIV positive people are already getting together and supporting each other. We do this over treatment literacy. We do this over just social support, how to talk to your family and your friends. And so linking, it's a good idea for any research group really to link into the community networks that are already established. And if you're lucky enough to be running some of the studies, in different countries, then build in a budget. So if you're running a research study, put in a budget for community engagement, include going out to talking to these networks that are already out there, maybe producing podcasts or information in other formats. And you're right, Cissy, everything we write is too complicated. You know, we want to take it right down. This is like you're having a discussion with your grandmother or something who doesn't really follow HIV research. You want to use everyday language, make it real in terms of what the implications are. So those two things might help. It would also help if all studies were listed with open information about enrollment criteria, and actually that they're going on. You know, even phase 1 studies should be listed on a register somewhere. Richard Jeffries, who's been involved in the IAS work before and is also on this webinar I saw logged in earlier, does a fantastic job of collating all the research that's going on. And so one way of publicizing it more is to know exactly what's happening. So those things might help.
Yeah, that's some great practical advice. And I might just mention that the IAS for the last 3 years have been running a program called, which is an academy for advocates for HIV cure research. They also run an academy, we also run an academy for researchers in cure research from low and middle income countries. Both of those are generally held in Africa. But the advocates one is excellent. It's sort of a lot of training about cure, a lot of training about handling media. And, you know, that's an opportunity. We usually take about 25 people each year and open process of applying. So please think about it. It's not just for advocates in Africa, it's for advocates in all low and middle income countries. So that's a great training spot. There's a question here, which I think is an important one, about what's being done around cure in people with co-infections like TB or viral hepatitis, given that that is so common, particularly in low and middle income countries. Debbie, did you want to speak about that, perhaps?
So I would just say I can only talk about the pediatric agenda for children. And I know there's a question in the chat around children and their participation in clinical trials. And the clinical trials for infants that we're working on and that has been in the field now for about 7 years is very early treatment of infants within the first 48 hours of life to replicate the case of the Mississippi baby. But because these include new drugs and new biologics, such as broadly neutralizing antibodies, kids with TB, et cetera, are excluded from those trials because of safety considerations of using new agents. And in general, for clinical trials, because they're novel therapeutics, co-infections, children across the age spectrum, even to young adulthood with co-infections are not included, active co-infections are not included in such clinical trials.
So are we going to change that, or do you think there's ways we can change that?
Well, I think we know how to treat TB. And so I think, certainly following treatment and, if we follow models of, say, cancer where suppressive therapies can be given for opportunistic infections, I certainly along the lines what Cissy was talking about, we have to think broadly and how to expand these trials to other populations. But at the same time, every adverse event that we get in these clinical trials come with a risk of stopping the clinical trials. And I think we have to proceed cautiously, but obviously when we have data to be able to think about how to roll out these in different scenarios safely. I think safety is still, I think the bottom line is antiretroviral therapy is effective, certainly in prolonging life. And we do have to keep that in mind as we proceed in this space.
Yeah, and maybe if I can make the point that participating in cure studies can be interventional studies, but also observational studies, just understanding what those co-infections do to the reservoir. And that was highlighted as a priority in the strategy for a cure, that we need to actually understand how those infections are interacting, what impact it might have, as a pathway towards one day doing interventions in those populations. There's another question I think is important. First of all, thank you to Jessica Saz-Weedle. Thank you, Jessica. I should have mentioned that the Cure Academies are a partnership between IAS and AVAC, and Jessica's kindly posted the link there should you want to come and join one of these academies. See, there's a question about the reservoir. Do all patients have a reservoir How do they form? And are reservoirs at all linked to virological failure? So these are sort of some conceptual ideas that I think are important for the community.
Yeah, so I should have defined reservoir. Reservoir is so when you go on long-term antiretroviral drugs and you suppress the virus, you can't see it in blood. There are maybe about 1 million cells in the body that contain the provirus, the DNA for that virus that can actually rebound if you stop therapy. So that's the reservoir, about 1 million cells, plus or minus. And it persists for decades, and that's why we can't cure people. So you know, it's hard to measure. It's the needle in the haystack thing. It's so uncommon, and to be honest, most of the time you find remnants of HIV in the body, it's dead stuff. The real stuff, the stuff we worry about, is pretty rare. Does it lead to virologic failure during treatment? No, actually. One of the most remarkable things in my career has been the fact that the treatments that we have now, the antiretroviral drugs, if you take them every day and you have a virus that's susceptible, that's a separate question. No, that reservoir is basically shut down and will be shut down for 70, 80 years, as long as you take these pills every day. And it's a reservoir that we're trying to get rid of, because if you get rid of that reservoir, there's no virus left, and then you're basically cured. Was that the question, Sharon?
Yeah, that's great. Thank you. Very clear. I think that would have cleared up some areas of confusion for some of the questions. Yes, Simon, go ahead.
I think maybe if you have a really high reservoir treated in really early infection, the clinical implications is it might take a bit longer to become undetectable. And that's quite often important to know by people who get upset that the viral load doesn't become undetectable within maybe 3 months or even 6 months. I think there's been a couple of studies that have showed that these people had a particularly high reservoir, and it's not going to cause treatment to fail. It's just going to give you a different timeline for becoming undetectable.
Thank you, Simon. Cissy, there's questions whether can transplants be done in low-income countries like Africa? And what you're talking about gene therapy, but is that going to be really expensive?
Thank you. Those are all very good questions. We know that today you can cure sickle cell disease using transplant. And for HIV, I think what they did really for the London and Berlin patient was transplant. But I think, as already Steve said, the transplant to cure HIV is very complicated. And what those patients went through, you cannot subject to people who are taking their antiretroviral drugs and they are okay. And actually, these patients only had these transplants because they had an additional complication of a blood cancer. So, but is transplant available in sub-Saharan Africa? There is, I think, some transplant that is happening in South Africa. Ghana started to do some transplant for sickle cell disease, but they stopped. Also, the infrastructure and training for transplant is complicated. Gene therapy, on the other hand, even if we are looking at ex vivo, would be much simpler than transplant. So transplant hasn't happened today in sub-Saharan Africa, but gene therapy will happen because it is possible. And also the optimum that we are going for is the in vivo. And I think many groups are really working towards getting an in vivo gene therapy. So the struggle continues with bold moves and bold decisions, including taking these gene therapies. And Debbie said, you know, we have to be careful. Everybody is so careful all the time when it comes. Can we take this to sub-Saharan Africa? No, just hold on. This might go wrong. if you don't try, you never know what will go wrong or what will go right.
Yeah, thank you, Cissy. I think we're unfortunately at time here, but maybe I'll use some of the words that Deborah said before, that these cases of transplantation give people hope. And I think that's what a lot of our discussions are here today. They give people hope, they inspire us that cure is possible. And now we need to make them simpler and more accessible and accessible. And that's one path is through gene therapy and through people like Cissy, leading programs there. That's a pathway to one day see that in the field. And I wanted to thank all of our speakers tonight. It's been really fabulous how many people are here and from all around the world. You'll be asked if you've joined the call to fill out an evaluation survey as you leave the webinar. I know that sounds really boring, but that feedback's really important to us so we can continue to make the webinars better in the future. Thank you so much for participating. There's clearly huge interest in this and from all around the world. It's just been great to see everyone join. The recording will be available on the Educational Fund website. And we'd also like to let you know that the IAS Educational Fund is organising another exciting symposium entitled Reflections on the COVID-19 Pandemic and its Application to the HIV Response in West and Central Africa. That will be on June the 1st and 2nd. So if you're interested, please register through the link in the chat. Hope you have a great morning, afternoon, or evening. I'm going to bed right now. It's 11:30 at night here in Melbourne, Australia. And thanks again for joining us and hope to see you soon.